There we go. All right, so we were encouraged strongly to keep our remarks short and to focus on one challenge to brain science. And so I'd like to just convey one bottom line message today. And until this morning, this seemed to me like an inspired and new message, but no longer. The message is that it's very important to recognize and know about different cell types in neural circuits if you're trying to understand how they work. I think in some ways this is a more serious challenge than the large number of neurons in the nervous system. We're often told that there's 10 billion neurons in the human brain, such a complex system. Science has developed tools to think about systems of many, many components. What we don't really have good tools for is heterogeneous systems of many components of several hundred different types. And I think that's where a big challenge is going to lie in trying to understand neural circuits. Now, I'm not at all an expert in discovering cell types or defining what they are. We've heard this morning from Nat Heinz, and we'll hear more experts talking about this problem. I'm more of a user of this information, and I've come over the years to recognize just how important this is if we are to make sense of neural circuits at all. And so I'd like to illustrate that with a few examples. The neural circuit I'm working on is the retina. The retina, as you know, lines the back of the eye. It's the first stage of your visual system. The rest of the eye is just there to project an image of the world onto the retina. The retina begins to process that image and then eventually sends information about that image onto the rest of the brain through the optic nerve. The retina, in many ways, it's kind of an ideal playground for neuroscientists trying to understand circuits because it just allows such exquisite experimental access. It has a very clearly defined input layer, the photoreceptors at the top of this picture. And you can stimulate 100,000 or so of these in your dish by just shining light on them. It also has a very clearly defined output, namely the retinal ganglion cells that normally send their axons through the optic nerve to the brain. And they're arranged conveniently in this lowest layer where we can pick up electrical signals with an array of microelectrodes, for example. And in between, there's a lot of interesting and complex circuitry, and we can manipulate that circuitry either by sort of classical means, sticking electrodes in and recording and injecting signals, or more and more by molecular genetic means, by modifying or eliminating certain cell types within the circuit and seeing what the consequences are. So overall, our goals here are to understand what it is that retinal ganglion cells extract about the visual image, what kind of visual computations do they perform. We'd also like to understand how the circuits within the retina perform these computations, so I don't really understand it as a machine, the way you might understand a radio. And finally, we'd like to understand how this function of these circuits relates to the needs of the visual system. What happens downstream with this information? Where does it go and how does it get used? So what about cell types? Well, there are 5 major classes of neurons in the retina. They're totally unambiguous. In this layered circuit, we have 2 synaptic layers, the so-called outer plexiform layer and the inner plexiform layer. And the 5 classes are defined just topologically by how they connect to these layers. The photoreceptors bring the inputs into this layer. Horizontal cells connect within this layer. Bipolar cells connect between the layers. Amacrine cells connect within this layer, and ganglion cells send the output to the brain. So these 5 subdivisions are really unambiguous, but if you look closely at the neurons in these classes, you find that they come in many different shapes and sizes. In fact, retinal anatomists have declared that there are on the order of 60 different types of neuron in this little circuit alone. And in fact, if we just look at the output neurons, the retinal ganglion cells, they seem to come in about 20 different types already. Now, each of these types of ganglion cell completely covers the retina, and so it really sends a separate image of the visual world to the brain. The optic nerve carries about 20 different images of the visual world onto the brain for further use. We'd very much like to know why the eye needs that many retinal ganglion cell types. But before I get into that, let's maybe step back for a moment and ask, how do we really know that there are 20 types of ganglion cells? How do we know it's not 5 or 100? Or as some people have said, just a continuum of different response properties that gets divided into classes by anatomists when they look through the microscope, but is fundamentally a continuum. Well, it turns out there is a hard and fast answer to this. It's not a continuum. There are discrete types, and the brain knows that they are discrete types, recognizes them as such, and the retina gives us the answer like this. It turns out that cells of a given type cover the retina in a regular array. Here's an example. These are the so-called alpha ganglion cells of the cat, and you can see that they cover the retina in this nicely spaced regular array. They seem to spread themselves out such that every point on the retina is within reach of just one of these ganglion cells, or maybe two. And generally, there's the impression that the retina tries to distribute its resources evenly so that they are used everywhere on the surface. Here's another example. We're looking now at two cell types. The starburst amacrine cells are in red, and another type of amacrine cell, the A2, are in green. And you can see that each of them forms an array of relatively regular, with quite regular spacing. Now, you might not believe that, so I have to show you the analysis. This is what was done here by Dick Maslin's group. What's calculated here is something called the density recovery profile. This is the probability of finding a neuron as a function of distance from another neuron of that same type. If the neurons were scattered about randomly, then this probability should be constant. You should have the same probability of finding a neuron no matter where you are. But instead, there's this defined hole here at short distances, meaning that you're very unlikely to find a green cell right next to another green cell. And similarly, for the red cells, you're very unlikely to find a red cell next to another red cell. But if you compare red and green cells, there's no effect. They're arranged randomly relative to each other. So there's a sense that the starburst cells recognize each other and repel each other. And similarly, the A2 amacrine cells have some form of repulsion, but the two cell types don't interact with each other. Now, how this happens developmentally on the retina is an interesting mystery. I'm not going to get into it, but people are starting to figure out the mechanisms that produce these arrays. We, just in a purely utilitarian gesture, can use them as a test of what is a natural cell type in the retina and what isn't. Now, of course, the same question arises everywhere else in the brain. It would be fun if there was a similar test that you could use, let's say, in the cortex for what is a natural cell type. And invariably, my friends say, oh, yeah, Markus, the retina, it's such a simple system, and you have this wonderful mosaic pattern, but that doesn't happen in the cortex. We just can't use that. But I wonder whether that's in part because we don't have sort of crisp enough cellular labels, cell type labels yet, with which to analyze these things. Now, a few days ago, an interesting paper came out in Neuron, very actually beautiful paper, that announced the creation of several new Cree lines of mice that direct the expression of anything, but in particular fluorescent label, to GABAergic neurons, different types of GABAergic neurons in the cortex. Now, some of these lines are clearly promiscuous in that you can show, in fact, the authors show, that they include neurons of different transmitter type and so on. But there was at least one construct here using a combination of two different drivers that the authors thought provided a highly selective expression pattern of GABAergic neurons in the brain. And I spent the weekend looking at these sections more carefully. In this case here, I chose the region of interest, which was the upper layers of the cortex, and just looked at the distribution of the labeled neurons in there. And lo and behold, they seem to repel each other. So if you look at the density recovery profile, there's a hole here with an exclusion distance of about 35 microns for this cell type. They seem to try to avoid being close to each other. And another line in the same publication showed an even greater exclusion distance of 70 microns or so. So this is a very crude early analysis from data that were not published for the purpose of this analysis. This can be done much better, obviously. These sections were 50 microns thick. What you'd really like to have is a 3D resolution, be able to see and measure spatial positions in the third dimension. But we were told this morning from Christoph how that's going to be done. It'll be done at the Allen Institute with these fantastic 3D microscopes. And so I think combine that with the fantastic resource of Cree driver lines, we'll be hearing much more about this topic sometime soon. Let me get back to the retina, though. So we have this catalog of retinal ganglion cell types, And this is just to emphasize that this happens in every species where it's been looked at, and in some cases there are also clear correspondences of types across species. But let's get back to the fundamental question: why does the eye use 20 different types to communicate to the brain? And again, answering this will require understanding what each of these types actually encodes about the visual scene and where that information goes downstream. Now, we've been helped tremendously in this by this sort of collective effort to find mutant mice, or generate mutant mice in which specific cell types are labeled. This is an example of several mice that were created by my collaborator Josh Sains. In each case, we're looking at a vertical section of the retina. The ganglion cells are down at the bottom, and you can see that one or a few are labeled green. And you can also see the arborization of their dendrites in the inner plexiform layer. And each of these types has a different pattern of arborization, and that is a consequence of their being different natural cell types that make different synaptic connections to the presynaptic neurons. Let me tell you what we can learn now by targeting these neurons selectively for electrical recording. Let's start with these JAM-B neurons. JAM-B is a mystery protein that for some reason marks a subpopulation of ganglion cells, about 5% or so in the mouse. If you look at an in situ stain here, you can see that they lie in a fairly regular array. And again, if you do the DRP analysis, it shows that there's this repulsion distance. So this identifies them as a natural cell type. Josh then went on to make a knock-in Cree line that allows the entirety of the neuron to be labeled fluorescently, and this gave us this interesting surprise. This is a picture now of a whole mount of the retina. You're looking down at the retinal ganglion cell layer. This is the optic nerve head. You see all the axons running across the retina to converge here where the nerve punctures out the back of the eye. And what was sort of immediately surprising about this picture was that the cells have these grotesquely asymmetric dendritic trees. They're not like all the cells I showed you on the previous picture that have nicely circular dendritic trees. Instead, they all point in one direction, and they're all extremely elongated. So this kind of thing was impossible to find without the genetic label. Asymmetric dendritic trees had been seen in morphological surveys before, and people always put them off as some kind of developmental aberration. You know, stuff happens. It's only through this technique that we were able to recognize that they were, in fact, a population that's sort of genetically identical. Now, obviously, we targeted them for recording and asked, well, what do they do with the visual stimulus? They turn out to be off-type neurons, so to a small spot of light, they respond when the light turns off. They also have a strong antagonistic surround to the receptive field, where the response is of the opposite sign of an on-type. And that surround was very asymmetric relative to the cell bodies. It was pushed in the direction of the dendrites. So obviously, we tried to see what happens when the spots move, and it turned out that these neurons were very selective for motion in a particular direction, and that direction, again, was the one in which the dendrites point, so down on the retina. And you can see that in response to a moving spot, there is no firing for upward moving spots and a lot of firing for downward moving spots. This is, again, downward on the retina. Remember, the lens in the eye inverts the optical image, so what it really means is upward in the world. So somehow the mouse has this one channel of vision that is dedicated to small dark things moving upward. Why that is, we really don't know at this stage. But we do know that this information gets transmitted down to higher centers in the visual system. These neurons project to both the thalamus and the superior colliculus, the two major targets of the retina. And in the superior colliculus, there are, as we know from classic experiments of Dreger and Hubel, it's a population of neurons that responds very strongly to dark spots moving upward. Now, at the time, in 1975, the conclusion was that this, what you're looking at here, is processing within the superior colliculus, because there was no knowledge that such responses existed in the retina. What we believe now is that the retina already computes this particular feature of the visual image and is just transmitting it more or less faithfully to this particular population in the superior colliculus. Let me tell you another story. This is a more recent insight. We've been working on this population of ganglion cells called the W3 ganglion cells. Again, we have here a a line, a transgenic line that labels them fluorescently. Now, these neurons had been seen in previous surveys. In fact, every survey reports them because they are the smallest ganglion cells in the mouse retina. And they also occur at high density. And so we expected that maybe these are kind of generic pixel detectors that just send the image from the retina to the brain for further processing in the brain at high resolution, a little bit like perhaps the midget system in our retina. Well, it turns out not to be true at all. These are already very highly specialized feature detectors, and they respond to only certain things in the visual scene. And let me tell you a little bit about that. First, again, if you start the simplest experiment where you turn a spot of light on and off, these neurons actually fire both at the onset and at the offset of the light. So they are excited by just about anything that happens within their receptive field. But if the spot gets a little bit too big, then they shut down entirely. This is the relationship of the strength of the response as a function of the size of the spot that you shine on. So once a spot starts to intrude into this, what we call the surround region of the receptive field, they actually stop firing entirely. In fact, it was difficult to make these neurons fire with anything interesting, like the kind of thing that a mouse might actually experience. So what I'm going to show you now is a little movie up here of what the mouse might actually see in real life. Here it is. This movie was taken with a video camera strapped to the head of a rat, and then we allowed the rat to go out and observe for us. We brought this movie back to the lab and projected it onto the retina of a mouse, and you have to excuse the mouse-rat difference here, but the Meisters can't carry the equipment. But the first line here is a record of the spikes from a W3 cell, not a single spike in a minute or so of stimulation. Whereas other neurons that are not of this type fire furiously and are obviously representing what goes on in this movie. So during this episode, which reflects a common everyday occurrence when the animal walks through its environment, the W3 cells are completely silent. So what is it that they actually signal? We tried hard, like I said, to look for ethologically interesting stimuli that they encode. And the only one we've come up with is a small dark object moving through the receptive field. So we played around with silhouettes of birds in the same way as Tinbergen did a long time ago. And it turns out that a small bird moving across the retina against a featureless background produces a big burst of spikes. So this is the spikes in the population relative to where the bird is on the retina. If the bird gets larger, there are fewer spikes, and when the bird gets too big, there are no spikes at all. So these neurons are quite selective for these objects as long as they're small, meaning at a distance where you can still run away. And once they get too close, the suppression from the surround silences them. So we think our best guess at this point is that they are specific detectors for small predators or predators at a distance. It kind of coincides with the fact that their density is very high in the inferior retina, which, as you remember, looks up in the sky. And finally, I want to emphasize that we're not the only ones in this business. So here, for example, is another cell type, the so-called PV5 cell that Boton-Roska has reported on recently. A beautiful set of experiments where they used stimuli that expand on the retina. So this is a time series here. Starts with a small bar, then it gets wider and wider. A little bit like an object that's looming and kind of coming close to you. So it expands on the retina as it approaches. And these neurons seem to be quite selective for this kind of looming stimulus. They fire strongly when a black spot expands, but they have no response to a bright spot or to lateral motion or to receding motion. So again, they seem to be quite specialized. And this somehow is a kind of a recurring feature. Now that we're able to look at individual types of ganglion cells selectively, we find that they are each quite specialized for one thing or another. And I don't expect you to make sense of what's on the slide. But just to say that each of these types seems to be driven by quite selective features about the visual image. And also what's interesting, and I'll leave for you to explore, in each of these cases, we can kind of define what the neural circuit is that actually produces these response properties. So there's been quite a bit of progress on that front as well. let me just leave you with a short note about the future. I've said that the third thing we'd like to explore is how this retinal code, the messages conveyed by individual ganglion cell types, is used downstream and what kinds of behaviors it drives. I mean, one extreme hypothesis about the 20 cell types is that perhaps each of them is evolution's answer for a particular behavioral need. And it's kind of a little module that then is used downstream, maybe in combination or so, but it essentially isolates from the visual image some feature that is necessary for a particular thing, a particular kind of behavior. And so we'd like to make these connections between cell types and behaviors. The biggest obstacle is that we don't know enough about visual behaviors of the lab mouse. We know they can be trained to do fantastic circus tricks, but we'd really like to know what is the natural repertoire. What are the kinds of things that they do in response to visual stimuli that perhaps evolution has shaped. So I want to show you a quick movie. We're just starting to explore this now, putting mice into an environment that is with a controlled visual input and see what happens. So what we're looking at here is a box that has a monitor on all sides, including the ceiling. We can't see the ceiling here directly. The box has a little cabana in it where the mouse likes to hide. It's more comfortable there, but every now and then it comes out and explores. And when it's out, then we start a visual stimulus and see what happens. We're going to start actually with a stimulus that's on the top, on the ceiling. And you can't see it directly, but you'll see a small reflection of it down on the bottom, on the floor that the animal's moving on. And we've also put this little mirror strip here so you can see it a little better. And the first stimulus is going to be an expanding dark spot on the ceiling. Later on, we'll see dark spots on the floor and also light spots on the ceiling, so I'll just play it. Okay, here you can see the dark spot on the ceiling. And she runs off into the cabana. Here again, dark spot on the ceiling, runs away. This is a dark spot on the floor now. And if anything, she's attracted to it. Comes back to see what's going on there. But very different, right, from the previous responses. This goes on for a while. Here we have a bright spot on the ceiling. Now, you might think, well, it's just the ceiling, something approaching from the top. And this went on for a while because we were really surprised that there's no reaction here. Doesn't seem to bother the animal much. Okay, she'll come back, you'll see. Okay, this is the same animal immediately thereafter with a black spot. Yeah. So there's a sense here that it's a behavior that's elicited purely by vision. You can't tell me that, oh, it's olfactory or whisking or anything like that. It's elicited purely by the visual system, and it seems to be quite selective in ways. We have stimuli that are quite closely related but have either a strong response or no response. Now, you'll also notice that this is somewhat related to the responses of these PV5 cells that Boton-Rosca described. In particular, a strong response to dark expanding circle and no response to lateral movement or a bright expanding circle. And we would love to know now whether these neurons that are genetically identified, whether they actually play a role in this behavior. And because I'm out of time, I can't tell you. Mostly because I don't know yet, but I'll be happy to tell you on a future occasion. So with that, let me just thank the people that have worked with me on that. Yifeng Zhang, who's now headed to Shanghai. In-Jung Kim, who is now at the Yale. Masa Yamagata, who is still with Josh Sains, a colleague in my department. Thank you very much.